At any rate, I want to welcome all of you here. Thank you very much for coming to the 24th Annual University Undergraduate Research, Scholarship, and Creative Activities Conference, which is a horribly long name for what is the Undergraduate Research Conference. So thank you very much. And I'm very proud of all your students and your children. And as a parent, I think it's really exciting to see your kids give a talk about their academic interests. It's a lot different than sending them off as freshmen or sending them off to kindergarten. They're suddenly adults, and that's a big change. So thank you for your support of your children, and thank the faculty. We've got Julian Malouf here, another faculty member here that's got a student who's presenting. So this research conference, as I said, has been going on for 24 years. This is the largest one. There's 533 students participating. 325 of them gave posters yesterday, 11 presented during the arts exhibit, and 197 are giving talks today. What they had to do to participate in this is they had to write an abstract, and that was judged, or they selected whether they wanted to speak or give a poster, and then it's compiled into a book, and I hope you got a copy of a book, especially if parents, be sure and get a copy of it. It fattens up the baby book, okay? So, the students are going to speak for about 12 minutes, and then there's an opportunity to ask questions. And all of you are welcome to ask questions. It doesn't matter who you are. If you're grandma, your aunt and uncle, your sister, go ahead and ask questions. What this is an opportunity for the students to say what they've been working on. They've been working very hard, and they've put together a presentation that describes their research and what the conclusions are, why they did it, and what the applications are. So we have 6 speakers this afternoon. The first one, I'm going to go ahead and start. Just for the speakers, I have a timer that I have set for 12 minutes, and I'll try and remember to flash the 2-minute warning sign. And no, that doesn't mean it's a 3-point after that. So it means figure out how many slides you still got to go, and let's get through it, okay? So there's water up here to help yourself. I also have cough drops because I don't usually sound like this either. There's quite a bug going around. So the first speaker today is actually from my own group. This is Mike Christie, and he is a senior. He just graduated, actually, and this will warm the hearts of all you parents too. He actually has a job, which is wonderful. So Mike is going to tell us about a ripening mutant of tomatoes and what its impact is on pathogens. So Mike, why don't you go ahead, and let's get you up here. And there is a slide changer and a laser pointer for you. Okay, and I will try not to drop the crowd's still in the water.
You ready? Mm-hmm. All right, good afternoon, everyone. I'd like to say thank you for coming. My name is William Christie. Like Ann said, I'm a senior, and I recently graduated last winter quarter. So 4 or 5 weeks ago. I majored in biological sciences with an emphasis in molecular and cellular biology. So I'm here today to discuss the project I've been working on, expression profiling of colorless non-ripening, or CNR for short, and its role in the susceptibility of tomato fruit to pathogens. Understanding the fruit-pathogen interaction is important to us because, as many of you know, we are slowly overpopulating this planet, and eventually food is going to become a very valuable resource to us. So understanding this interaction, we hope to minimize the loss of fruit due to rotting. So an overview of the points I'm going to discuss in this slide include the model plant pathogen interactions that we observed, susceptibility and the ripening process, CNR and its role in preventing susceptibility, the experimental strategy we use, and the results and conclusions from this experiment. Excuse me. So the model organisms we chose to work with were the tomato fruit and the gray mold detritus scenario. We chose the tomato fruit not only because it's important to us as a source of food, but because it's been used for a long time as a model organism to see the ripening process due to the fact you can see the ripening steps fairly easily. The tomato has had a genome sequence, so it makes molecular studies a lot easier. and provides molecular tools in the form of mutants, specifically ripening mutants, which we're interested in. We chose Botrytis cinerea because it's one of the most common forms of fungal infections in fruits worldwide. It is also an ideal model organism for necrotrophs. In other words, it feeds on dead or dying flesh. Like the tomato, it has had its genome sequenced and provides the same molecular tools in the form of mutants. So this slide shows the normal ripening process of a fruit, or tomato fruit, as it goes from mature green to red ripe. As I said earlier, you can see the stages pretty easily. Mature green, breaker, pink, light red, and red ripe. For the remainder of this presentation, we're only going to focus on the mature green and the red ripe forms of ripening. So these fruit were infected with Botrytis cinerea and allowed to mature for 3 days. As you can see down here, the mature green fruit, sites of infection, remain relatively resistant to the pathogen. But if you look over at the red ripe fruit, you'll see that it becomes susceptible to this pathogen. There are many factors that affect the fruit's ability to ripen normally, and these do include specific genes. The gene that we're interested in, however, is CNR. CNR, or colorless non-ripening, regulates the tomato's fruit ability to ripen normally. As I mentioned earlier, a normal ripening in the simplest form goes from mature green to red ripe. The CNR mutant, however, does not let a fruit ripen normally. As you can see, the mature green is called mature green-like because it is the mature green of the fruit, but it doesn't quite look like an actual mature green fruit, as well as the red ripe form. In fact, they're slightly colorless, giving it the name colorless non-ripening. So, following the logic I presented earlier that mature green fruit are resistant and red ripe fruit are susceptible, you would assume that because CNR mutant fruit are unable to ripen, that they would remain resistant to pathogen infection. This, however, is not the case. Like I showed you earlier, wild-type fruit that have been inoculated 1, 2, and 3 days past infection still remain resistant. The red ripe fruit, 1, 2, and 3 days past infection, show susceptibility. Now, we infected CNR mutant fruit with the Botrytis cinerea pathogen and notice that it has a hypersusceptible phenotype, meaning that it's susceptible in both the mature green and the red ripe stages. As you can see, the mature green stage is right here and the red ripe over here. Some of you may think that the red ripe actually looks ripe. In actuality, it's quite literally been obliterated by the Botrytis infection. So from the last slide, you can see that CNR does affect the normal ripening process in a fruit. This slide, however, presents the question, is CNR involved in the resistance of tomato fruit to pathogens, and if so, how? We believe that it's the expression level of the CNR that plays a role in this fruit's resistance to pathogen infection. Now, our strategy to test this hypothesis is we first collected mature green and red ripe fruit from both wild-type and CNR mutant plants. We inoculated them and left some of them healthy to use as our control. From these healthy and infected samples, we proceeded to extract the RNA and synthesize cDNA. With this cDNA, we performed qPCR analysis to see the expression levels of the CNR gene. Excuse me. So this graph shows the results of our qPCR analysis. Each of these bars represents the relative expression levels of CNR in both the mature green and red ripe fruit in both wild type and CNR mutant fruit. Now down here you'll notice it's 1 and 3 DPI. DPI meaning days past infection. The reason we chose 2 different points of infection is because we wanted to see if and how the expression levels of CNR changed throughout the ripening process after being infected. So what I'm going to explain to you first is, if you notice in the green bars, those are all the wild type healthy fruit. If you notice, the expression levels stay relatively similar throughout the ripening process that goes from mature green to red ripe. Now, if you look at the red bars, you're going to see those are the CNR healthy fruit. While they display the same type of relative, the relative amounts being the same, you notice that the expression levels compared to the wild type are significantly decreased. This, in fact, is what the mutation causes, a significant decrease in the expression levels of the CNR gene. In the previous slide, I posed the question, is it involved in tomatoes' resistance to pathogens? This graph actually shows that it is, and I'll explain that to you right now. So, moving on to the good stuff. Looking at the wild type infected, you notice that there is a massive increase in the initial infection of the fruit with the Botrytis cinerea pathogen. If you go back to the mature green, you remember that it's resistant. Now, if you look at the CNR infected fruit, you remember that it had a hypersusceptible phenotype. This shows that the increase in the CNR gene is actually what provides that resistance. And you can also tell because, remember, as I mentioned, when it went from mature green to red ripe, it became susceptible. You can see that in the infected fruit, the actual levels of CNR decrease. Now, if you look at the red ripe fruit, which I mentioned were susceptible, you'll notice that there's no initial increase in the CNR expression levels, but it still continues the trend of decreasing. As far as the expression levels in the CNR, they remain the same as they were before, decreasing. Oops, looking at this. These results tell us that the CNR gene expression levels do in fact play a role in a fruit's resistance a fruit has against pathogen infection. So, in conclusion, CNR does aid a fruit in its ability to ripen normally as well as resist infection to pathogens. The mutant form of CNR inhibits the ability to normal ripening, and it also inhibits the ability of a fruit being resistant to pathogens, thus making it susceptible to pathogen infection. Now, the study of susceptibility is interesting to us because not only does it show the dynamic interaction between pathogens and fruit, but it also has various applications. Understanding this interaction we hope, will lead to a decrease in rotting fruit or being able to minimize rotting fruit, giving us a higher crop yield. Another important point was that it was previously thought that the ripening process and a fruit's ability to resist or be susceptible to pathogens went hand in hand. So as a fruit ripened, it was going to become susceptible no matter what. This shows that that actually is not the case, that there are different pathways that can be utilized. The advantage of knowing that is that eventually we hopefully will have fruit that can ripen normally without being susceptible to pathogens. And of course, you have to give credit where credit's due. I definitely would not have been able to do this without Ann Powell, my sponsor and PI. Barbara Blanco, who's actually one of my good friends and mentors. She's not here today, she's actually overseas. Kalai, who happens to be right here, one of my other good friends and colleagues. And Stephania, she couldn't be here because she's actually working on her thesis. So, thank you everyone, and I hope you enjoyed my presentation.
Okay, questions for Mike? Does anybody want to know about rotting fruit? Does anybody have yes, go ahead.
I was going to ask, is that gene, the CNR, is that part of a defensive signaling pathway or anything, or is it just an individual gene that you've isolated?
It's an individual gene, but there are also other genes that affect ripening. It's just one that we've honed in on to see, or that we've found to show expression levels having something to do with resistance. Did that make sense to you? You want me to?
No, that's fine. So
okay.
Okay.
So yeah, we haven't exactly mapped out the pathway. We just know that that is one specific gene that is involved in ripening and a fruit's resistance to pathogens.
Yeah, Julian?
So how would you use this knowledge to engineer or perhaps breed for tomatoes that did have more resistance later in the ripe stage?
Well, we probably have to map out the entire pathway because, I mean, it's not as simple as one, okay, this gene definitely does this. I mean, we haven't mapped out the whole pathway. All we know is that from what we've seen, it appears that it does affect resistance and susceptibility, but there could be many different forms, other things that it affects. So ideally, if we could find out what this is and map the whole pathway, then we could engineer some fruits that are going to be resistant to pathogens in the future.
I was wondering about expressing CNR at higher levels.
Oh, you mean?
Throughout the ripening stage or something. Do you think that might work?
It's possible. I haven't looked into that, but it's definitely possible, and that makes sense that that's how it would work. So.
I don't think that's been done yet. Do you know what the mutation is in CNR?
Oh, so the CNR mutation actually is a hypermethylation in the promoter region of the CNR gene. Hypermethylation meaning there's a whole bunch of methyl groups attached to the cytosine groups. This prevents transcription factors from binding to that promoter, leading to the decreased expression levels.
So it's an interesting gene to look at. So, for other reasons too. Other questions? Yeah.
Has anyone ever looked at other fruit? Maybe there's an analog of CNR that is implicated in ripening of other fruits that people look at?
I'm not 100% sure on that. All I know is that we've looked at tomatoes, but I haven't seen any other research done into other fruits. Do you?
I'm not aware of anything. It's a SBB binding protein, so I'm sure it's present in other fruit, but it's not as well characterized. Yeah. Other questions? Good. Thank you, and thank you for a good discussion.